Assalamualaikum and hi. In this video, we are going to explain about serological technique, which is ELISA. So, what is ELISA? ELISA is the acronym of enzyme-linked immunosorbent assays. It is a serological technique in which this technique is used to measure the presence and concentration of antibodies in the blood, and it focuses on proteins made by your immune system. About ELISA. ELISA is also known as a heterogeneous enzyme immunoassays (EIS) technique used in clinical analysis, used to detect the presence of antigens in biological samples, in which the samples used in ELISA include serum, plasma, cell culture supernets, cell lysates, tissue lysates, saliva, and urine. Next, there are four principles of ELISA. The first one is the term sorbent. It is antigen of interest is adsorbed onto plastic surface. Second, the term immuno, antigen is recognized by specific antibody. The third, the antibody is recognized by second antibody, immuno, which has enzymes attached, which what we call enzyme link. The last principle is substrate reacts with enzyme to produce product, usually colored. And then we have types of ELISA. There are four types, which are direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. The first one is direct ELISA. It only uses one antibody, and the antigen or sample is added directly on the plates, and a conjugated detection antibody binds to the target protein. Substrate is added, which produces a signal that is proportional to the amount of analyte in the sample. Next, indirect ELISA. This ELISA helps detect the presence of an antibody in a sample, and the steps is identical to direct ELISA with additional amplification detection step. It requires two antibodies. A primary detection antibody binds to the specific antigen, and secondary antibody complementary to the primary antibody. Substrate then added will produce a colored product. And then we have sandwich ELISA. It is the most common type of ELISA. It helps to detect the presence of antigen in sample. It is being called sandwich because the antigens are sandwiched between two antibodies. Capture antibody is coated on a microplate. Sample is added, protein of interest binds and immobilized on the plate. And then the detection antibody is conjugated and facilitates the detection of the antigen. And then substrate is added, which form a colored product that can be measured. And then we have the competitive ELISA. It is commonly used for small molecules. It helps to detect antigen concentration in sample. It is kind of identical to sandwich ELISA, but instead of use detection antibody, a conjugated antigen is used to compete for binding with the antigen present in the sample. The more antigen present in the sample, the less conjugated antigen will bind to the captured antibody.
What are the procedures of ELISA? The first step is an antibody is attached to a polystyrene plate, which is a solid surface and is attracted or has an affinity towards the bacteria, other antibodies, and hormones. The second step is a microtiter coated with an antigen is filled with this antigen-antibody mixture, after which free antibodies are removed by washing. Third step is a second antibody specific to primary antibody is added, which is usually conjugated with an enzyme. Next, the free enzyme-linked secondary antibodies are removed by washing the plate. And the last step is the substrate is added. The substrate is converted by the enzyme to form a colored product, which can be measured by using spectrophotometry. For the four types of ELISA, which is competitive, indirect, sandwich, and direct ELISA, we can see the similarity in their procedures. The first similarity that we can see from the procedure is the first step, most of the antigen and antibody are coated. The second similarity is in the third step, enzyme conjugated antibody will be added to the antigen. And the last similarity is in the last step or the fourth step, substrates will be added. The substrate helps to develop the color of the antigen or the antibody.
Next, we move on to the advantages of ELISA. Firstly, easy procedure. It is easier to perform it as it comes with a simple procedure. Second one will be elevated level of specificity and sensitivity. It is more accurate for better result. The third one will be high efficiency. It is capable of performing simultaneous analysis without difficult sample pre-treatment. And the next one, cost effective. It is because of the cheap cost of reagent. And furthermore, safety-wise and environmental friendly. It is not necessary to use large amount of organic solvent or any radioactive substances. The last point will be ELISA is based on antigen-antibody reaction. Next, we move on to disadvantages of ELISA. Firstly, it is expensive and labor-intensive to prepare an antibody. The second one, it is necessary to use sophisticated methods and costly cultural media. The third point will be high risk of false negative or positive result. It is because of the high sensitivity and high specificity. The next point, instability of antibodies. The next one will be since an antibody is a protein, it must be transported and stored refrigerated. So it is quite a hassle. And the last point for the disadvantages of ELISA. False results arise from insufficient blocking of immobilized antigen.
Everything was created with a function. Now, I want to explain what function of ELISA and how it's worked to human. As we mentioned earlier in video, ELISA been used to measure the presence and concentration of antibody in the blood. First of all, ELISA been used widely in the world, especially in clinical lab, because the function of ELISA that used in clinical analysis. It used to measure our antibodies antigen by our blood, saliva, or urines. It works because ELISA assay rely on enzymatic reaction between an enzyme conjugated label and substrate to produce a colorimetric, chemiluminescent, or fluorescent product. Other than that, ELISA also really significant due to hot issue that is COVID-19. As we know, during 2020, world being affected due to COVID-19, so ELISA or serology testing playing a big role to us. Based on my friend that been affected COVID-19 experience, first of all, a professional health care will take a blood sample usually by finger prick or by drawing blood from a vein in this arm. After that, the blood sample was tested whether you are develop antibodies against the virus COVID-19 or not. If the results show you have antibodies, that means you have been infected with COVID-19 in the past or you being vaccine. Now, I want to explain how it's worked to detect the antigen and antibodies. ELISA serology is usually done in multi-well microtiter plates, so that is a dilution of plants. To perform that assay, the wells of the plate are coated with the antigen of the interest. In commercial test is done by manufacturers. To begin the test, the wells are filled with dilution of the patient blood. If the antibodies against the antigen are present, they will bind with the antigen fixed to the bottom of the wells, but only antigen-specific antibody will bind to the wells. After that, the well will wash out to remove all unbound antibodies. After that, the solution of animal antibodies against human antibodies will be added. Then the second antibodies will covalent conjugated to an enzyme. After that, the well will wash again and unbound enzyme conjugated antibody will remove. At the end, solution of colorogenic enzyme substrate is added. The development of color in the wells with a specific antibody can be seen with the naked eye or under microscope. To conclude, ELISA is really function to us for sustainable life. Thank you.
To conclude, ELISA or enzyme-linked immunosorbent assay is a laboratory technique that detects certain antibodies, antigen, and other substances in blood or body fluid. This technique is known as gold standard of immunoassays among researchers. It is also known to be one of the easiest blood tests that can be carried out because the process is rapid, quick, and only needs a blood sample of the patient. It is a very important test in diagnosing infectious disease by examining the presence of antibodies in the body and offer various advantages and disadvantages in different aspects. And that will be all from us. Thank you.